Well, thank you everyone for joining. Sorry, we were having a little bit of technical difficulties, so it took us a couple extra minutes to get started. But my name is Josh Morrison. I am the president of One Day Sooner, and I'm really just thankful for everyone for joining, and really thankful and honored for our panelists to be here. It's really true that we have literally some of the leading experts in COVID challenge studies in all the world talking to us today on this panel. And so we're very excited to be hearing from them. I would be a little remiss in my position at One Day Sooner if I didn't take this opportunity to just kind of really briefly tell a little bit about the organization, what we've been working on these past few years, or these past few months. And One Day Sooner is a group that represents people who want to be in human challenge studies. And we've had a really busy and exciting 2021. Besides our work on human challenge studies, we've also done some exciting work on vaccine equity, sponsoring Vaccine Day celebrations in Boston and Springfield, Massachusetts. And we have another one upcoming, hopefully in El Paso in the spring. And we've also worked with the West African Health Organization to create a petition to the World Health Assembly to have a vote of all the world's nations on a pandemic insurance fund. And that letter has been signed by 15 different West African countries in the economic community of West African states that represent 350 million people. And that's been led by our One Day Africa chapter and its chapter director, Zach Kafuko. In the next year, we're going to be promoting challenge studies beyond just COVID-19. And, that work has already started with some work already that's really exciting on tuberculosis, where Gabrielle Kleinwax, our communications lead, published a paper yesterday in Vaccine on the kind of economic case for tuberculosis challenge studies. And A.B. Rohrig, our senior policy advisor, has a paper under review at the American Journal of Bioethics with some of the folks at Oxford on the bioethics of TB challenge studies. And we're really eager to be expanding beyond COVID and to be thinking about, how to use challenge studies for diseases like group A strep and influenza, possible hepatitis C. And we have an event coming up in January in DC, which will also be on Zoom on January 12th about, it's going to be a debate on would it be ethical to do wild type tuberculosis challenge studies? And I really encourage you all to be doing that, to be joining that. We have a number of projects that's going to be coming to fruition in the first quarter of next year that we're really excited about. We're going to be able to announce an exciting new grant for our organization, and we'll be publishing a report on the history of COVID challenge studies that has been compiled of dozens of interviews with some of the leading players in the field, and that's been led by Mabel Rosenheck. And we'll also be publishing a joint statement from leading experts on all sides of the COVID challenge debate about that supports creating a standard of pre-study protocol publication for all COVID challenge studies. So kind of look forward to seeing that. And we're hoping to make that kind of the standard for challenge studies and hopefully other clinical studies in the future. But the last thing I want to mention before introducing our moderator is a project that we've been working on for the past few months, which is to create a compendium of all the challenge studies that have been launched in the last 5 years and that are currently kind of ongoing or that have been announced. I'll put the link in the chat. And really, One Day Sooner wants to be of service to the challenge field in general and be trying to provide these kind of public goods that make it easier for people to run challenge studies and make those studies more effective. And so this compendium, which is kind of in its kind of early kind of beta stage, was put together by our Chief Operating Officer, Julia Mirza. It includes about 150 studies. We'd love for everyone to take a look at it and see, particularly if there's anything that we're missing, if there are studies that are there that we haven't identified yet, because we really want this to be a comprehensive list and a really useful tool for the whole field. So with that, I want to introduce our moderator, Dr. Nadine Rufael. And, I first want to say when I started One Day Sooner, almost 2 years ago now, back in March of 2020, I knew basically nothing about vaccinology and basically nothing about challenge studies. And when I was first researching, learning about challenge studies, I remember reading Nadine's systematic review of influenza challenge studies, and I saw the name at the end, Nadine Raphael, and thought, oh, like I desperately want to talk to this person. This is such a powerful, useful insight. And so it's been amazing getting to work with Dr. Raphael and having her here today. Nadine is a physician scientist with expertise in all phases of clinical trials for vaccine, diagnostic, device, antimicrobial resistance, epidemiology, human challenge models, and translational immunology studies. She has experience in human challenge trials relevant to respiratory and enteric pathogens. Over the past 12 years, she served as the national chair or co-chair, or as well as the lead PI or site PI of 50 different protocols, and as a co-investigator for more than 130 trials addressing a variety of infectious disease priorities in diverse populations. She serves as the executive director of the Hope Clinic, the clinical arm of the Emory Vaccine Center, and as the Emory VTEU PI since 2018. She's also the principal investigator of the critical care for the NIAID-funded Emory Human Immunology Project Consortium, where she serves as the national chair for its clinical subcommittee and as a member of its scientific steering committee. We're thrilled to have Nadine moderate today's discussion. I'll turn things over to her.
Thank you so much, Josh. It's such a pleasure to be here and be among giants in challenge studies, either from the bioethical perspective or the implementation and conduct perspective. I'm also very glad that Dr. Eyal was able to join us despite some technical challenges at first. So thank you. This is really my pleasure. I will go ahead and introduce our famous panelists for today. I will first allow Dr. Eyal to comment a little bit more about his most recent paper accepted. Then we will have it open to the panelists so they could share more their experience and perhaps ask additional questions. I will take on a few Q&As that I will ask Dr. Mimoli and Dr. McShane and Dr. Eyal to comment on. And any of your questions are very welcome, so please go ahead and put them in the chat box. So it is my pleasure to introduce Dr. Nir Eyal. He's the inaugural Henry Rogers Professor of Bioethics at the School of Public Health. He's also the director for the Center for Population Level Bioethics, Institute for Healthcare Policy and Aging Research. He's also an associate faculty member in the Department of Philosophy, School of Arts and Sciences. And his work is in global health, mostly focusing on health inequalities, health promotion, and research ethics. He engaged many bioethical challenges, including healthcare rationing in resource-poor setting, critical health worker shortage, disaster response, universal health coverage, and ethics in HIV and emerging infection trials such as COVID-19. I also got introduced to One Day Sooner through Nir roughly back in April 2020 when I read his first paper about what could COVID vaccine challenge offer as a solution to the pandemic. It's also my pleasure to introduce Professor Helen McShane. She's the director of the Oxford NIHR Biomedical Research Center. She's a professor of vaccinology at Oxford University, deputy head of the Medical Science Division, and an honorary consultant physician in infectious disease. She researched a new vaccine related to TB, and her group is currently studying whether delivering a TB vaccine via the aerosol route is more effective methods of vaccination compared to the traditional route. In the past few years, started clinical trials of this aerosol delivery of the vaccine candidates and have shown that this route is both safe and immunogenic. Other projects include a BCG challenge model in humans, as well as understanding the role of helminth infection on vaccine-induced immune response and the evaluation of protective efficacy of new antigen and viral vectors. So welcome, Professor McShane. Last but not least, Dr. Matt Mimoli from NIH. I also had the distinct pleasure of shaking hands with Matt and meeting him in person before the pandemic in the Bethesda campus. Matt is a world authority in influenza challenge in particular. He also used it as a way to understand human influenza pathogenesis and correlates of protections, and as well as understanding emergent infectious disease through the use of a challenge model. He is currently serves as the chief for the Laboratory of Infectious Disease Clinical Studies Unit at NIH. So welcome everybody, and I'll go ahead and start with Neil. Thank you.
Thank you so much, Nadine. Can everybody hear me and see me in the slide deck? Yes? Fantastic.
Yes.
Thank you so much. It's such a pleasure to join you, and I apologize for the technical issues figuring the program. This is a joint paper with Abby Rohrig from One Day Sooner, and we got funding from NSF and Open Phil, and Abby worked for One Day Sooner. I advise for free. This is basically our conflicts. And it was published in JID not long ago, before Omicron, but I think I'll try to apply the claims there also to our new situation with Omicron. We may need to develop strain-specific vaccines in response to emerging variants, potentially including Omicron. We'll know much more in a few weeks. How do you test, how do you develop them? And specifically, how do you test them in human subjects as opposed to just testing how things work out in the lab? To determine efficacy in humans, regulators in the US and in Europe have said that it would be enough to rely on clinical immunogenicity studies. But we don't absolutely know what the immune correlates, we suspect strongly, but we don't know for sure what the immune correlates of protection are. And that leaves some uncertainty about the results of an immunogenicity study. And I can imagine an environment in which the FDA, which has been criticized in past months for being too conservative on various grounds, might say we need more human testing of one kind or another, if at all possible, to appease also the vaccine skepticism in the public that has risen since that time, and whatever other considerations that have motivated the conservatism in the past. And Abby and I propose that if additional efficacy data in human subjects are deemed necessary, there are reasons to use challenge trials in this context. They could furnish a lot of the necessary data and do it in an ethically acceptable way, we argue. What roles might challenge trials play? Well, the relevant central role would be to establish efficacy. Specifically, challenge trials would be good for establishing the efficacy in reducing infection rates, infectiousness rates. They're not a good tool for trying to gauge the impact on disease and especially not severe disease rates. Basically, because of an ethical advantage, we hope that there would be very little, if any, incidence of those things in these trials, given the selection of young population. Challenge trials, human challenge trials, I'll shorten to HCTs, could also uniquely help to determine various other facts about the disease. Specifically, everybody agrees, including the opponents of challenge trials that I'm citing here, that challenge trials are a particularly useful tool, better than field trials, conventional phase 3 trials, to establish what the immune correlates of protection are and give us that information more reliably than we have so far. In Britain, they have been preparing, I think maybe by now have prepared culture for examining the Delta variant in challenge trials. For newer variants, that could create delay to create the culture in the onerous conditions typically required for conventional challenge trials. In that way, I'll mention that Mark Lipsitch and I have proposed a special sub-design of a challenge study that can circumvent that need and basically do the exposure without having created virus culture. To try instead to do the efficacy testing in conventional phase 3 field trials would simply not work now, I believe, for a variety of reasons. First, there would be complications. There could be around the study site declining case rates. This disease, waxes and wanes around particular environments in ways that we can't completely foresee in advance. There might be interaction of different strains complicating the analysis, and it might just mean that the researchers then need to say, okay, we need to start from scratch in a new site. Such things have happened in COVID field trials in the past. If the design envisaged is a placebo design, what we're talking about now that vaccines are widely available is you ask people in great numbers, tens of thousands of people potentially, don't take this vaccines that are important for your health and important for the non-consenting people around you. So jeopardizing in a serious way the rest of the community, which never gave us consent to experience that risk. If we're talking about an equivalency design that compares not anybody to placebo, but different vaccines to one another, these require even more study subjects than we have seen in the placebo field trials that we've seen in the past, basically to establish the difference, the small difference between two potentially efficacious interventions or known to be efficacious interventions. The question is just which is more efficacious. You need a greater number of participants, and that's just, or more time passing, and that's just making it all non-realistic. And besides that, there are also complications in getting the number of vaccines that we need for, say, that equivalency study in the great numbers needed. Maybe that's something we could resolve for a small efficacy study like a challenge trial that recruits maybe 50 to 100, maybe 150 people. We just can't resolve that for these tens of thousands of people, I believe. So you might argue it would have been fantastic to do challenge trials for the sake of making a dent at the global pandemic. The social value is pretty clear if you just think about the multiplier, how many lives are lost every day to this pandemic, much greater social value, even given some uncertainties than in most clinical trials that we approved. But you might say, yeah, but say Omicron is most probably very transmissible. It's a ethical problem in that way. But of course, if the participants are isolated as they are in the dose escalation study that has ended in Imperial College in London, and as they would be in any challenge trial, then the transmissibility has zero impact or almost zero impact on creating any risks for the study subjects who are anyhow going to be exposed to the virus, or on the community, which is isolated from them. So this really does not pose a special ethical challenge. We don't know, but some future variants, maybe not Omicron, maybe yes, Omicron. The hopeful sign so far is that Omicron is not that kind of variant, but maybe Omicron and maybe others will be more lethal, and that you might think does make it very problematic to expose human participants deliberately to a variant that is going to be even more lethal than the variants that we have dealt with so far. But we believe that the risks in challenge trials that have been as much as they were characterized so far are so low compared to the correct bioethicists' upper thresholds for how risky a clinical trial can legitimately be, that we are well within the threshold, the zone of acceptable trials if we do that. There are assessments. I see that Mannheim is with us today. Great honor. Their most reliable analysis so far suggests a risk that is of 0.00025% of death for the individual study participant. And even if you encounter a strain that is ways above that, it couldn't possibly be at the level of the more dangerous organ transplantations, live organ transplantations, which doctors regularly approve. I note that Frank Miller is participating today. It's a great honor. He came up with this ingenious way of proposing what is the highest threshold for risk in clinical trials. And Frank and his colleague, Steve Joffe, said, this is for routine medical trials. In situations like pandemic, you might actually approve even more dangerous studies than that. But even so, this is an example of something that doctors regularly approve, not for the donor's own sake, but of course, it's not good for the donor medically to donate a right liver lobe. It is something that they do for other people's sakes. Some of the comparisons were to less dangerous voluntary organ donations. I think that was a mistake. That's misleading. We need to compare it to the most dangerous live organ donations. The doctors still approve when patients provide their voluntary consent. There are a few differences when you donate. Typically, it's for somebody who's already right now an identified individual in some senses, even if you don't know them, that is in great need. Maybe that somewhat elevates the urgency, but I just think that the numbers in a pandemic, the social value is so tremendous that it easily balances from an ethical point of view. And overall, the social value side is extremely important for almost whatever findings we can have about the disease or about the vaccines. So it easily justifies the level of risk for the individual participant. Finally, we increasingly have more effective therapies than we had when the debate about challenge trials exploded in spring 2020. So thankfully, maybe now we have, I don't know if we should call it the rescue therapy that some have sought as a condition for conducting challenge trials, but we can decrease the risk. And this is what is important. In fact, from my point of view, what really matters ethically is not so much whether we have a therapy or not, it's whether the risk can be low enough. And the findings from Manningham and others already established that the findings are probably low enough. There are some open questions. We don't know, for example, most centrally, whether the level of exposure in the studies is similar to the level of natural exposure that was observed by the studies that Manningham and al looked at. But still, it's just hard to imagine the numbers for accidents being anywhere in the ballpark of what we don't prove if the difference between 0.4 and 0.00025 is so large. So I think that there is ethical license. And of course, the studies would need to be ethical in a variety of other ways, fully informed consent, rigorous informed consent, kind of serious study participant selection, etc. But they could be ethical. So my conclusion is that boosters which are strain specific, if they are needed for Omicron, or some think it's just a matter of time till we need something like that for other variants, could be tested if needed through challenge trials, which would also provide other important information, and that testing could be ethical. Thank you.
Thank you so very much. This was great. I don't know if Helen and Matt would like to comment in particular about Nir presentation or just in general about COVID-19 challenges before we started our discussion.
Hi, it's Helen. Can you hear me?
Yes, and we can see you too.
Finally have sorted out my IT, so apologies for that. So thanks, Neil. That was a really nice introduction. I was particularly pleased that you said that you thought one of the things challenge models were particularly good for is understanding correlates of protection. I completely agree. And that's the rationale and the basis for the study that I'm leading in Oxford at the moment, where we are enrolling subjects who've been previously infected with SARS-CoV-2 with a very explicit aim of trying to identify correlates of protection. So I agree with that. I guess the only other thing to say is that, I mean, of course, most of the use of controlled human infection models has been to test vaccines and will continue, I expect, to be so. But actually, these models are equally suitable to test other therapies, monoclonal antibodies, antivirals, and potentially diagnostics as well. So I think, you know, I am an infectious disease physician by training and have been, as Nadine said, developing a tuberculosis challenge model for the last 5 years. And I think in infectious diseases, we have not yet fully exploited the use of controlled human infection models.
So I will jump in as well. I agree with what Helen just said. I think it's very important that we think beyond just testing vaccines, and that these kinds of studies are actually not just a short-term sort of thing to do, but should be part of a long-term plan for studying respiratory viruses. Because what some of us have known for a long time, because we've been working on this, and what a lot of people are learning throughout this pandemic, is that these types of respiratory viruses are very complex. And what's going on during an infection with immune responses is very difficult to understand. And so these types of studies can be very useful as part of a long-term research plan to understand correlates of protection, transmission, questions like original antigenic sin, and so on and so forth. I mean, I could sit here and talk about it for half an hour. So there's a lot that can be done using these kinds of studies. The other thing I wanted to say was I was pleased to see you talk about the risk, because one of the things that I think has been misunderstood by many during this pandemic is the actual risk to an individual of death from COVID. And if you're a young, healthy person under 50, that risk is extremely low. And in the setting of a challenge study where you're talking about people that you've de-risked up front, where you're screening the patient, making sure they're completely healthy, putting them in an ideal situation where you're monitoring them, that risk drops even further, as Mannheim pointed out. So, you know, all that, and to me, the important de-risking is up front in that screening. It's not, do you have a rescue therapy or a drug? It's are you screening properly? That's where the de-risking happens. So it's important that we understand that risk. I mean, the risk to a person under 50, even in the wild, is 0.001% of death in the, you know, most of the time. And that's lower than your risk of death on the highway in the United States anyway, which is 0.0125%. So, I mean, we really have to put things in perspective in terms of risk.
Yeah, absolutely. Great point, Matt. It's really about risk and benefit. We talked a lot about the benefits through Neil's presentation, and you guys have also added that it's not just trying to test new vaccine candidates, but also getting correlates of protection, better diagnostics, additional therapeutics. I guess my question, beyond de-risking and the availability of rescue drugs, which seemed to be a point that a lot of people had focused on early in the pandemic, where we did not have access to antiviral or monoclonal antibody or immune modulator. But can we also talk a little bit about the risk of long COVID? I mean, how does that come into play? Not really the mortality aspects of things, but the, you know, more on the morbidity side.
So, yeah, I mean, I'm willing to jump in and say a few things about that. So, you know, we know for a fact that with many different infectious diseases, there are individuals who develop long-term complications. In many cases, we don't understand. So, right, you can find a person who's had traveler's diarrhea, they come back and their GI system is just never the same again. There are people that have had influenza and they develop some weird, you know, autoimmune type syndrome or some issue that lasts many years or for the rest of their life. And these happen, and you could call them long influenza, long COVID, long, travelers, whatever you want to call it. These absolutely happen, and they're real, and they are things that we don't fully understand. But there are also individuals who maybe go through a difficult hospitalization, or they go through a difficult period with the virus and they become decompensated, and they develop various symptoms related to either the treatments that they underwent, say positive pressure ventilation, or just decompensation because they were laid up for weeks, and they're a little older, and now they are tired and they can't exercise the way they did, or they can't work the way they did. And some of that is also being called long COVID, when in reality, it's decompensation or related to treatments. So we have to be careful. And then there's the third sort of category that I would put, which are, there are people who develop rare symptoms or unusual symptoms. You want to call it chronic fatigue syndrome, you want to call it other autoimmune diseases that people develop. And sometimes they decide to blame a recent infection, which may or may not have had something to do with it. And there was a study, I don't have it on the tip of my tongue here where that study was, but I know I read a study, I believe out of the UK, where they followed a group of infected and non-infected people looking at long COVID symptoms. And in many cases, they saw the same rate of symptoms in both groups, demonstrating that a number of the things that are being called long COVID are actually just random issues that people are developing health-wise throughout. So what I'm trying to say is that I believe that the number of people that truly have long COVID, they exist, but they're much smaller than the total of what's being called long COVID. And so we should be careful when we think about long COVID in discerning those groups. That's not to discount that becoming decompensated and having complications of treatment from COVID isn't important. It is, but that's not really a risk in a challenge trial, if you see what I'm saying.
Yes, thank you. Helen?
Yeah, hi. So agree with absolutely everything that Matt has just said. I think what we call long COVID quote is a huge collection of very different things, and we're throwing them all into the same bag, but I think it is a really important point, and it certainly was something that we discussed a lot in terms of setting up the challenge studies. I think the challenge, and no pun intended, was that we didn't at the start know what the clear risk factors were for this for this syndrome. There are now some clear risk factors that have been defined, although of course the detail is less clear, and as Matt said, there are lots of different flavors of what's called long COVID. I think for me, though, what this really comes down to is the fully informed consent process as well, and ensuring that you are very clear with participants that there may be a risk of this. To a certain extent, that is difficult to quantify, but absolutely documenting that you have had that discussion with a participant is an essential part of the informed consent process for me. For our study, it was a little simpler in that, of course, we were enrolling people who'd previously had COVID, and part of our inclusion criteria were people had to have been fully recovered for at least a month prior to our reinfection and had no enduring symptoms or signs of any sort whatsoever. So in a sense, I mean, I have not seen clear data on this, but the risks of quote long COVID after a second infection, when you have not had it after the first, I think are very substantially lower.
Thank you, all great points. Nir, go ahead.
From an ethical analysis point of view, the long COVID factor is interesting. It's in a way the mirror image of what we were talking about earlier about lethality. So lethality is the most severe event, but it's very rare. With long COVID, the worry is that while relatively mild or moderate, whatever forms it takes, it might be a collection of several different possibilities, but all of them are in that range. Altogether, it might be fairly common, and then you have, what do you do with the ethics of that? Is it okay to save, help save with some uncertainty, with only partial support, contribute to the global fight against a scourge that has killed 800,000 Americans and so many millions of people around the world and continue to kill and devastate economies and thereby indirectly devastate lives and health. To do that, when you know that there is a risk that, which is more substantial probability-wise, that somebody will have maybe even not mild, maybe moderate harm as a result. We don't have a full ethical theory about that, but I think it's worth it. Some colleagues have said, I think some people who have the honor of being joined by today's call said, anyhow, the numbers about lethality are bumping against that upper risk threshold. So the long COVID factor might just kind of push us above that threshold. But if what I said earlier is right, we are nowhere near that upper risk threshold. So they don't do that.
Right. No, thank you all. And I think, one important thing is there's just so much we don't know yet about long COVID, and just it's such a huge spectrum, as Matt alluded to. So as we find out more, we can also be able to quantify the risk even better. I know we have a question from the audience. So Danny, if you don't mind unmuting Seema Shah. Great. Fantastic.
Hi, can you hear me?
Yes.
Great. Thank you. This has been really interesting. So I was I think one of the things I struggle with is the question about the time it takes to develop a strain for a challenge study, and how one thing I learned from Matt Memley is that in a way with the development of new variants, we're always going to be in this process of playing catch up. And I think what I was hearing from Nir is what I hear from Matt is maybe the best way to think about it is just have a longer term strategy where you're looking at multiple strains and maybe even developing a pan coronavirus vaccine, which I think makes a lot of sense to me. But from Nir, I think the response about getting access to the benefits of challenge studies more quickly was, well, maybe you could do an infection where people are exposed to other people who are infected with this new strain. I think that's what you said in your paper with Mark. So that leads me to kind of 3 questions. I'll say them very quickly because I don't want to take up too much time. So the first is, I'm curious from Helen and Matt, if you use this approach of exposing people to people who are already infected, what does that do for the things that you care about, trying to learn from the challenge studies? Does it make it harder to find correlates of protection or test treatments or vaccines? And then with the new variants, I'm curious whether we would know enough, or when we would know enough about the level of risk and how well our existing treatments actually can deal with those variants. And then finally, with the social value, my view is based on Annette Arid's work. I think it's really important to be precise and clear about what the pathway to social value might be. So I'm curious to hear from Helen, who's been doing this work, what's the best case for the social value that challenge studies provide, even if you don't have a challenge study with the variant that's currently the one that's circulating and causing the most concern. So thank you.
Thank you. Helen, please go ahead.
Hi, yeah, shall I kick off on that? So I think the idea of doing, it's a sort of hybrid between a controlled human infection model and a natural history study if you're doing a deliberate exposure study, which I think is what you're describing. So sort of the household contact type environment. I think that is a hybrid with some of the advantages and some of the disadvantages of the two on either side of it. So it is less controlled than a controlled human infection model. You're not, in our controlled human infection model, we are inoculating direct into the nose. So there is no question that the virus isn't in the nose. So if a person is not infected, it isn't because they haven't come into contact with the virus. I think the limitation of even a household contact type setting or a deliberate exposure setting is you can never quite guarantee that the person has been exposed to enough of the virus in the right place, as it were. So, but obviously that kind of setting might be a smaller kind of study than a huge, 10,000 subject phase 3 trial. So it's, I see that as sitting in the middle somewhere. It's a bit of a halfway house, and I'm not, you know, my preference would be to do a proper controlled human infection model, because then you've got the tightest model, so you can do it in the least number of subjects, because you've got less statistical variability in terms of whether or not people have actually been exposed. Sorry, the lights go off in this room if I sit still for too long. So if I start rocking, it's just because I'm trying to turn the lights on. Your question on new variants. So at the moment, the studies that are underway are obviously using the Wuhan strain. I think for me, when we started these studies, the huge advantage of that was the strain we knew most about clinically. And I think the sort of minimum lead time for manufacturing a strain to GMP and doing all the adventitious agent testing is about 4 months. I think actually that's okay in my mind, at least, and I'm happy that other people may think differently, because actually I think it takes you that long to be confident. I would feel, how can you take informed consent with a new variant when you don't have enough clinical data to know what the consequences will be? I think, you know, informed consent does depend on you having some information. And I think the less information you've got, the less rigorous that informed consent is, and the less comfortable I would feel administering an agent. So for me, that is okay. And then I wasn't quite sure what you meant by your last question about value.
I think we need. Go ahead.
Okay, thank you. Yes, so my question was, you know, the question about social value, I think the way that Nir was framing it was anything that helps with the COVID pandemic, because we've had so many deaths, would be clearly very valuable, right? And if there's even a low probability of something helping with such a, you know, high potential benefit, then you just do that calculation, multiply probability times that benefit, and you're done. Other people have argued you kind of need to be more careful about what's the pathway. How does a challenge study contribute to value? Is it something where you identify a correlate of protection and that speeds up future vaccine testing to have a proxy endpoint? Is it that you're testing new treatments and rapidly prioritizing treatments for testing down the road? And, you know, my question is, when you think about trying to catch one of the newer variants and doing this, but you're always a little bit behind, what's the best case for the actual value and the pathway to population benefits that you get from doing a challenge study? And, you know, given your experience, I thought you might be a good person to talk about that.
Sure. I mean, I guess I think the answer is we don't know yet, because we haven't finished the study and we don't have that data. And I think history will tell us how valuable these challenge studies have been. But I think the best case scenario is that we can establish some information on immune correlates of protection. I don't see challenge studies as standalone. I think those challenge studies complement information we get from natural history studies and confirm information we get from natural history studies. So we're not doing these studies in a vacuum. I actually increasingly think that if we can establish a human infection model where we can use it to test antivirals and monoclonals, that would be a really useful thing to do, because we can then down select which ones get put into large phase 3 trials for confirmation. And you know, you can have a kind of triaging process. So I think, and of course, you know, if we can, well, you can test new variant vaccines with a challenge model, depending you've got the homologous strain, but you can also identify immune correlates. So potentially you can bridge not only to a challenge, but to a correlate in terms of approving those new vaccines. So those are the key high-level things that I think we can contribute to knowledge on with a challenge model.
Could I make a couple of comments? So I'm happy to see Seema. We've worked on a number of projects together, and she characterized much of what I've said and think correctly, in that I look at these as long-term projects, as I said before, and I do think that, you know, broadly protective vaccines is where they could be particularly helpful. There's a couple of things I wanted to say about the variant issue in general. So what we have to remember is that a virus like SARS-CoV-2 or influenza does not exist in a human host as a single virus that all has the same sequence. You have a collection of virions that actually have different sequences, but there's one dominant sequence that makes up what we call the strain, whether that's the Delta or the Omicron or the Wuhan, right? But that sequence is not the only one that's there. There are others. And this is constantly changing through this process that we call intra-host viral evolution. And then that goes on an even larger scale within a population where the virus is changing because of the lack of error checking. And we're all familiar with this at this point. And pressures from the immune system, pressures maybe from drugs or vaccines. All of these things are causing the virus to constantly evolve. So what I'm trying to get at with this is that although, yes, it's true that we're always behind and we're never going to get ahead of the virus, it's just never going to happen. Overall, the SARS-CoV-2 variants are all still SARS-CoV-2. They are going to have fairly similar properties in terms of pathogenesis, transmission, correlates of protection, and even disease. Now, there could be some subtle differences, and it could become a little more infectious, and this and that. But a lot of that isn't really the virus. It's the interplay of the virus with the immunity in the population. So when we start to say things like, oh, Omicron seems to be less deadly, well, that's not really Omicron on its own. It's how Omicron is interacting with immunity in the population. So it all goes together in this sort of one big milieu of immunology and virology kind of coming together to give us whatever it is we have. Because if everyone had immunity to SARS-CoV-2 when the virus emerged, however it emerged, we wouldn't have had a pandemic, right? No matter how deadly the virus was. So we have to take all of that into consideration. So how does this apply to challenge studies? Well, I do believe that doing challenge studies with viruses that are not necessarily the latest and greatest variant can still be very useful. And even using the original Wuhan strain can tell us a lot about pathogenesis, transmissibility, correlates of protection, and so on and so forth. And then if you're trying to study vaccines that I'm particularly interested in, broadly protective ones, you actually want to have the older strains available because you can use them to help you interrogate whether these vaccines are broadly protective while natural history clinical trials are going on with whatever the current variant is out in the world. So that's where I see it as a benefit for vaccines. And as for social value, I think what Helen said is right. But overall, it's really, we should think of this not just in terms of SARS-CoV-2, but as RNA respiratory virus pandemics in general, because we've now had outbreaks of SARS, MERS, a pandemic with SARS-CoV-2. There's a likelihood, and we believe in the past there have been other coronavirus pandemics, just as there have been influenza pandemics. So it's a high probability that at some time in the future, whether that's in our lifetimes or in a future, generation's lifetime, there will be another coronavirus outbreak. And what we can learn now can certainly help in the future. Now, it may be hard to see for us, but we do have to think about that. And in science, we always think about the fact that maybe a lot of what we do won't become important until we're dead, but that's part of the game.
For me, Nadine, to add a word as well.
Yes, please.
Thank you so much. I just wanted to, I guess, 3 points. One is to say that SEMA is putting her finger on one of the most important challenges. A lot about the value of challenge trials to address specifically new variants will depend on the timeline and how much we could act fast enough or not. If one of the main advantages of challenge trials is speed, then we're in this, we by the time we were done preparing for Delta, we've got Omicron, etc., and it's a chronic process, then a lot of the value goes down. So it is very correct that it's a very fair challenge. About the option of trying to cut time by a natural exposure challenge trial, where you basically deliberately put a person or persons in a state of acute infection verified to be from the relevant variant in a room doing some activity that typically we know increases risk of exposure with a bunch of other people. That, by the way, could be done with multiple people in acute infection, so that you really have a very high likelihood to address one of the points that Helen mentioned, that there is exposure, that one of the people has exposed the others in the room. Regarding that design, Helen mentioned some disadvantages compared to field trials, but she also mentioned but did not name some of the advantages, and there are also some scientific advantages. The central ones is my understanding that if there is exposure, is likely to be at the typical doses of natural exposures, which are, after all, the kind of target thing that we're trying to tackle. And also the route of infection is less artificial than this, put stuff in your nose. It's how people get infected. And if the vaccine addresses that, then we eliminate one source of uncertainty. So there are also some scientific advantages. Helen mentioned the issue of informed consent. She said it gives her, she's actually not so concerned that we need to wait some time, because meanwhile, information gathers about the new variant and the quality of the informed consent increases. I want to say about that while perhaps the quality increases, it's not like at the beginning when there is relatively high uncertainty, although it's a new variant of a familiar virus. But certainly even initially in SARS-CoV-2 infections, I was inclined to say there can be informed consent. It need not be a violation of informed consent. What the investigators need very honestly to tell the participants to verify that they comprehend is that we don't know much. But that could be okay for informed consent. I'll give you an example. In the field trials that everybody, for the vaccines that we approved, that we authorized, there was exactly a situation of that kind. There was a first-in-human vaccine. We've never had vaccine mRNA and specifically mRNA for this disease before. It was never tested in a situation that could have brought out events like severity enhancements in human before that trial. And investigators were supposed to at least convey very effectively to the participants, there might be this new thing. We don't know. We don't know how common it will be. We don't know how bad it will be if it happens. Take it or leave it. And this is, I think, what people should honestly propose here, since those trials were surely ethical, or at least could be ethical if they indeed convey that. I'm not so sure that it was always conveyed. I participated in some sessions where it was not conveyed, where people were trying to recruit subjects. I feel that the challenge trials where the eyes of everybody will be on the investigators, and the quality of informed consent with a smaller number of participants is likely to be very rigorous and very high. I think it could be done.
Thank you, Nir. I know David had a comment. Maybe David, if you can unmute, or perhaps we have to do it for you. Thank you.
Go
ahead.
I was unmuted. So I want to comment, before my work at One Day Sooner, my dissertation was actually on value of information analysis and public policy. And one of the tricky things that we have for, like, how useful is a trial going to be? And so the first piece of research that One Day Sooner did was actually on this, was trying to evaluate use cases for HCTs for COVID. And there were a lot of things that they can be used for, as the panel knows. But I think what our review of past trials has shown, and what I'm sure everybody's experience is that when you do a trial, you often know what it is you're looking for, and you also learn other things. You can't justify trials because, hey, maybe we'll learn something. You know, the process of planning out exactly what it is you're going to learn from a study is obviously integral to designing the study. But I also think that it's a little bit difficult to say, well, we need to have decided before we've even designed the challenge study what it is that we're going to get out of a prospective future challenge study so that we can decide whether or not this class of trial is ethical. Because you actually have to evaluate this specific study and think about what it is that could come out of it, rather than talking about, and generally, a notional trial of Omicron, which doesn't actually describe anything.
Thank you, David, for the question. Neil, you want to go ahead and answer?
I didn't quite get, David, whether, so is the upshot that people are expecting too much of challenge trials, or that it's actually not okay that, so in my slides, I said, you know, you could learn a variety of things and one should be much more specific. So it's a sort of critique of challenge trials. From which side do you want us to answer?
Yeah, my point was just that the discussion so far has been about using challenge trials and whether they're justified. And I was just noting that challenge trials as a class can't be justified because of what it is you learn from challenge trials generally. A specific challenge trial can be justified on the basis of what it is you're going to learn from this specific challenge trial. So I think that if we have generally understood ethical principles for when it is that you can allow such trials, then you can start talking about this trial would be ethically justifiable because we think we can learn these things. And you might be surprised and learn more than that, or you might find out that actually you didn't learn the thing that you needed, because not all trials work exactly the way you want, but you do need to be talking about a specific trial.
Totally agree, and I'll just say the claim was challenge trials can be ethical. So there might be a particular trial that has no value, or is too risky, or is, you know, they fail to obtain informed consent or whatever. It's about the possibility. Sorry, Matt, you were.
Yeah, no, I was just going to say, I think David's hitting on something that I've pointed out to some people in the past as well, is that when you're doing challenge studies with a particular disease, as you're doing them, you should always be reevaluating whether what you're doing is ethical. Does it make scientific sense? Is it medically reasonable? Is it important? Does the benefit continue to outweigh the risk? Because, you know, the risk can change, the benefits can change, what you're getting out of a study can change. Maybe you've done enough studies and you've learned what you need to learn, and there isn't much more benefit to be gained for the risk, or maybe the risks have changed because the population has changed in some way. Or, you know, or as I pointed out in the beginning of the pandemic, at some points during this pandemic, it might, in certain places, it might have been inappropriate to do a challenge study because you would have been taking away resources from, you know, nursing resources, PPE resources, other types of resources that you may have needed to care for patients. So at that moment, it might not have been appropriate, but 6 months later, it might have been. So you always have to be reevaluating what you're doing. And also scientifically, like when I started the flu challenge trials, I have certain viruses that I've tried that I can't get a good infectivity rate. Well, I don't think it's worth using those viruses in future studies because I'm not getting an infectivity rate that can give me any power to study anything, but others do. So you have to always be reevaluating what you're doing. And I think that's sort of where David is going with this.
Yeah, no, thank you, Matt. It's clearly a iterative process. And I think just to add also, you know, encouraging people to have their protocols online, their ICF online with very clear, you know, objectives and endpoint is absolutely critical to the transparency of the process, for sure. And sometimes, you know, moving science forward could be also coupled with testing products, whether it's diagnostics or preventive or therapeutics. Helen, please go ahead.
Yeah, no, hi, just to say that I agree completely. I think David's point is spot on. And of course, this is why, you know, our ethical review process approves a specific trial. It does not approve a blanket, you can go and do some SARS-CoV-2 challenge studies. And not only that, but it approves specific versions of the protocol too. So as you amend a protocol, as you generate data and you amend a protocol because you want to do something, you know, you work on the basis of that data and change something perhaps that you're doing in the trial, that amendment also needs ethical approval before you can go forward. So it's a very tightly controlled process based on the specifics of what it is you want to do. And there was a question that Randall posted in the chat, which said may be useful to define the desired endpoint COVID-related studies. Well, absolutely, you'd never get a study through ethics unless you had a very clearly defined primary, secondary, and tertiary endpoint. Absolutely, these things have to be very specific and necessarily so.
So David, maybe one last comment from you and we'll go to Claire. Go ahead.
Yeah, just to kind of go back to, I think, what Nir had been saying. Yes, you have to approve one specific trial and you can't talk about them in general. But the other side of that is that you have to have a clear claim that this class of trial can be ethical, because trials are very expensive and nobody's going to even design a protocol unless they think that there's, a clear consensus that, yes, this class of trial can be ethical. And I think that that's the piece that you do have to have consensus about. And you have to have, people say very clearly, here are the types of qualifications that you need. And yes, Pfizer, we think that you can use this type of trial to check your next mRNA vaccine if there's another pandemic, because otherwise it's not going to be used and it's not going to speed anything up.
Yes, thank you. Claire, please go ahead.
Thank you. My question was about sort of pressing on this point about benefit, about whether we have any way of quantifying benefit from these studies, or a threshold for benefit, like we've talked about with threshold for risk. Because I think the sort of counter argument to say the transplant and living donor for transplant risk threshold is that you can also say it's clearly going to have a benefit and quantify that benefit. And I wondered if there was any experience of benefit thresholds that you thought applied to challenge studies, or even the ratio between risk and benefit. And, is there any way of quantifying the other side of the equation a bit more accurately?
Maybe the experts on what you can get out of challenge trial should answer. I'll just brief it, but I'll just say, from a decision theory point of view, there are some decisions in life that that are rational to make because we know that we'll have a benefit. But when the potential benefit is very high, it can be very rational to invest money, say, even if there is high uncertainty, even if you don't know what kind of thing will work out of the many good options. And so, and I think that potentially, so even if we don't hear very promising things from Matt and Helen, we could still be in that zone of hit space investing, or in Chef's terms, throwing spaghetti at the wall.
Yeah, so with scientific research, you're never going to have the same clear, cut benefit like you're talking about in that transplant situation, right? Where you have a person who's going to die if they don't get a transplant. You do the transplant to save their life. I mean, this is all very clear and clear cut. Whenever you do research, studies fail, things don't work. Things don't work out the way you expect. I mean, lots of, things can happen. So, there's always some less clear-cut research. I mean, even with a vaccine trial, right? How many vaccines just don't work? Or drug trials, they don't work, right? So you do lots of things where they don't work. Now, obviously, you could argue maybe there's a difference because you're taking certain risks in a challenge, but I would argue a first in human, if something never used, may even have more risk than giving someone a virus that you've watched for a year or two infect people, and exactly who's at risk and who's not. There are lots of benefits you can get out of challenge studies, and I think the key thing to remember is the difference between studying the natural history of a virus or a vaccine in the wild compared to a challenge study. And I have this slide that I show when I give talks all the time that has sort of two columns that show the differences. And the issues you have in natural infection studies, or even just looking at epidemiologic data, a lot of the data that we're fed about COVID is just not very accurate because of these factors. you don't have any control of the people that are in your two groups. So, when we look epidemiologically at the vaccinated and unvaccinated, well, that's fine, but it doesn't mean these are equivalent people unless we're making the effort to make sure that those groups are equivalent. And that can be very hard when you're talking about a virus spreading in the wild. You can't guarantee that a virus is going to infect this 50-year-old who's healthy and that 50-year-old who's healthy and this 25-year-old with a heart condition and that 25-year-old with a heart, you don't have that ability to control that. But in a challenge study, you do have the ability to control the population that's in your study. In the wild, you don't have the ability to control the virus. So you could be comparing someone who was in the Delta strain, the Omicron strain, some other strain. With flu, we have a few different subtypes we deal with. In a challenge study, you can guarantee that everyone is exposed to the exact same virus, genetically identical if you want to, if you generate the virus yourself. You have the benefit of knowing or even controlling what the immune profile looks like of the people going into the study. So you can look at an antibody titer, a T cell activity, whatever it is you want to look at, or many things. And you can say, hey, these are the people I want in my study, people who are naive, people who have, like Helen's done, previously infected, people that have certain immune responses. You can control that in a challenge study. You have no control over that out in nature. So when you're testing a vaccine or you're giving a drug, you don't know how many people did the vaccine work for, not because of the vaccine, but because they already have some level of natural immunity, or because genetically they're less predisposed to developing disease. There's all these different factors that you can't control that you can in a challenge model. Now, of course, it's still a model. And as Nir pointed out earlier, it's not the same in terms of infection, mechanism of infection, right? We're artificially putting it in the nose rather than natural transmission. So there are limitations, but you have this very controlled setting that allows you to do detailed immunology from the timing of the pre and post infection it's impossible. It's absolutely impossible to do in the natural setting. It's just impossible, because when you see a person with COVID or flu, you have no idea when that infection happened. It could have happened hours ago, days ago, a week ago. No matter when they tell you the symptoms started, you don't know when it actually started. And to me, that's really the key thing, is that's the key benefit, is having that opportunity to control those factors that are just impossible to control in the natural setting.
Thank you, Matt. Helen, please go ahead.
Hi, Claire. Just to say, I think very good question. The real challenge for me in your question is the risks are to the individual, whereas the benefits are to society. And I think I don't have a way that you can quantify that benefit, as we've all discussed. I think, you do a study with the best of intentions, expecting that it will provide data that will be useful. I see it as contributing, rather. I don't see this as binary. I'm confident that we will get data from these studies that will contribute to the global burden of knowledge, or the global volume of knowledge, and contribute to the development of new therapies and new vaccines, understanding protective immunity, etc. etc. I don't even know how you quantify that. But I think the key thing for me is that benefit is not necessarily to the individual, and the risks are to the individual.
Yeah, thank you. I love the discussion regarding risk and benefits. I'm just gonna shift gears just a bit to talk maybe about resources, wish lists, bottlenecks. What is it that you would like as leading the fields in human challenge trials? What do you think are the bottlenecks? Is it GMP requirement? Is it biocontainment capacity? Is it the acceptance of the community for those types of studies? Helen, I'll start with you, if you don't mind.
Yeah, sure. No, that's fine. Well, it obviously depends on what study you're doing and what pathogen you're doing. The SARS-CoV-2 challenges we are doing in containment, so in negative pressure rooms. So obviously that is a rate limiting step, although to be honest, it has not been a significant rate limiting step for us to date. I think necessarily because there is a very rigorous informed consent process involving two stages, a telephone screen and then a face-to-face screen, and a whole long list of exclusion criteria. Actually, for us, one of the rate limiting steps is volunteers who meet our inclusion criteria, actually, and who are eligible. And I don't see any way around that, really.
Ma'am?
So this is a interesting question. So, you know, I've been working on challenge studies for about 10 years now. And, you know, one of the things that has been very limiting for me in terms of influenza, but I think this could apply to some degree with SARS-CoV-2, is the lack of ability to, so I have a limited facility with a limited staff. I can only do so much. Right, I can only do so big a study with so many people, and there's been a lack of ability to expand and get connected with others who could do this. So in other words, I have a fairly good history now of doing these studies, kind of having some concepts of how to do them. I also make viruses myself, and we're pretty good at that, at making GMP viruses. In fact, I have about 7 or 8 different influenza challenge viruses that I've made, many of which have never been in humans because I don't have the capability to get that done. And so I would like to see more, and I know there are facilities like Emory that could potentially do these things, and many that are willing to do these things, but I haven't found a good mechanism by which to get all of us connected to work together to do these. And I feel that if I could get connected with good clinical teams that can do challenge studies, we could get a lot more done. And had we had that capacity set up before the pandemic, we might have been able to use that capacity to move a little bit quicker, at least in the US. I know in the UK, they've done a pretty good job of this, but to move a little bit quicker on working out whether we could do some of these SARS-CoV-2 challenges, or even other coronavirus challenges. I mean, I've made I'm in the process of making an OC43 challenge virus, which could also be used as part of a challenge study in which to test, say, a broadly protective vaccine or better understand coronaviruses. But even that, I right now, the unit that I use at the clinical center is completely being used for COVID testing, and I can't do any challenge studies right now. So if I had access to other groups that did have the ability to move forward, I could work with them to get some of this stuff moving forward. And you know, and there are groups that aren't good at making viruses. They can do a clinical study, but they don't know how to do these other aspects that I do know how to do. So I would like to see more opportunity to connect, collaborate, get funders to bring us together, and be able to work together to get a lot of this work done, because I'd love to see a number of my flu viruses, some of these coronaviruses, get through challenge units and get studies done, which would build the capacity for testing vaccines and drugs and so on and so forth. So that's what I'd like to see happen.
Can
Yeah,
I just
absolutely.
come back to that and say I completely
Yes.
agree. I think a network of challenge-ready sites, you know, and it might be quite a small network, but actually where there is focused expertise. I mean, we've had sort of similar conversations in the UK. There are about 5 or 6 sites which have across the UK that have experience in challenge studies. And I agree. I think because then you can expand, then you can do the numbers, then you can do multi-site studies, and you can push the numbers through quicker.
Yeah, absolutely. And it's beyond the US and the UK, with multiple biocontainment units across the world that could be tapped into. Nir, could you comment more on could we do things differently as far as raising awareness for challenge studies, better education for the community about what those types of studies are? Would you like to comment more on this?
I mean, I'll also take the opportunity to answer the original question. A pandemic can be much worse than COVID is. A human engineered virus could be much more lethal. In a way, COVID is giving us an opportunity to see how we should prepare and how we fail to prepare. And because of the response was so bad on so many levels, in so many countries. There was not you know, in terms of non-pharmaceutical interventions, in terms of the unnecessary politicizing of the crisis, in terms of the little scientific comparison of what interventions work and what don't, the waste of an opportunity to randomize interventions and learn more. It's really a window to how we should do things differently in future crises, which would also require a very fast response. And if they are worse, in many ways bolster the case for using radical interventions like challenge trials. So there is a lot that we can do now to try to remove barriers. I've just corresponded in the chat with Helen about the issue, the paucity of volunteers who can still pass through all the rigorous screening that they are doing for challenge trials. What about people from outside the UK? It's not practicable to recruit people who come in, stay in an hotel, then fail at some point of the screening later on. But the scale of the pandemic is such that it clearly is economically worth it to cough up more hotel rooms and more flights and do that. And there is no ethical problem with recruiting from another country. So there's so much that we could do, and we should plan now. If we are now starting to think about future preparedness, absolutely doing challenge trials with a lot of thinking outside the box about some regulatory matters like this one that we've just mentioned is really necessary, and would, ready centers, ready procedures, thinking very seriously about how much we do need GMP conditions for the culture and other issues that we should address now.
Yeah, that's wonderful. I just want to go back to a point that Matt had made. So, we've been talking what Nir had said, like we really need challenge models for new variants. Helen had said, yeah, but let's work with something that we know. We know the clinical disease, we know what works against it. Maybe a step back. I mean, is it too late to consider a seasonal coronavirus challenge? So things that have been done in the UK many decades ago, could they help us in trying to plan, maybe not for SARS-CoV-2, maybe for SARS-CoV-3 or upcoming? Did the wagon pass on doing additional work with seasonal coronavirus?
No, I think we absolutely should work on things like OC43, HKU1, even alpha coronaviruses, because remember, these viruses are all from the same family. They have a lot of similar properties. In fact, I think a lot that we've learned from influenza applies to SARS-CoV-2 too. In fact, I feel we've ignored a lot that we knew about influenza, and we shouldn't have. But these are RNA respiratory viruses. A lot of the immune responses you're getting are very similar across the board, especially when you look at things like innate immunity, cellular immunity. How these things work are going to be very similar. So I think this is a very easy, safe type of thing. And the other thing that it gives you is a window into the future of SARS-CoV-2, because it's highly likely that as immunity builds in the population, especially from children as they get older, how SARS-CoV-2, as I mentioned before, SARS-CoV-2, if it hangs around, is going to continue to evolve along with the immunity in the population, right? And 20, 40, 50 years from now, things look a lot different when everybody who's 50, 60, 70 years old had COVID when they were young or exposed to it when they were young, as opposed to now where you have, 50, 60, 70, 80-year-olds who never saw this virus before. So, looking at how these coronaviruses are behaving can really tell us a lot about the future of the new ones that we have, as well as just generally understanding them and potentially understanding how our immunity to other coronaviruses could impact future coronavirus outbreaks. And so I think there's a lot of questions, scientific questions we could ask surrounding this, even potentially doing sequential challenges with a seasonal and then a SARS-CoV-2, or a SARS-CoV-2 and then a seasonal. I mean, there's a myriad of questions that we could ask about transmission, about pathogenesis, about immunity, and there's a lot to think about. So again, we need more people to help us do all this stuff if we were going to even consider it.
Right, absolutely. Any more questions from the audience before we close the session? Well, thank you so much. This was really an engaging session. This was very, priceless input from the board expert on human challenge studies. And I think we have one more question from Bryce. Go ahead, Bryce.
Yes, so the discussion of challenge trials and relative risk is a little frustrating to me in that for SARS-CoV-2, we have a nearly 100% chance that any potential challenge trial volunteer will be exposed to it. It's very different than, say, Ebola, a rare disease that only a few people may be exposed to. So how can that factor into the ethical study? You're either going to be exposed to COVID-2 at the supermarket, or you're going to be exposed in a controlled environment.
Shall I jump in on
Yes,
this?
please go ahead, Neil. Yes.
I think you're totally right. That should factor in. It factors in, especially with Omicron, we have this unusual experience that people can get reinfected. But if you knew that the infection in the trial would save you from getting infected in a less, or is much likelier to save you from getting infected in a much less controlled environment, then it absolutely should come into the calculation. And people have defended challenge trials on that ground, both in earlier work about using challenges more in endemic environments in developing countries. That was one of the arguments. And I actually have a paper in ethics and health research that says once you take that factor into account, that further reduces how much risk we should attribute to participating in the trial. Because really, I think you're right. What does it mean to increase somebody's risks? Is to increase their risk compared to how they would fare otherwise. That is how, tort law or common sense assesses how much I put you at extra risk that you wouldn't have experienced otherwise. So fundamentally, you're absolutely right. I didn't mention that today because for some reason, a lot of people don't like taking that into account. In research ethics, people like to focus on the risks in the procedure, not so much compared to the background risk. There's some ways to try to defend that, but I think they fail.
Having talked about this and thought about this a lot, I would be a little careful about this. So there is, you sort of can consider it different when you're giving someone a virus on purpose that you made or generated in the laboratory, compared to, even though it's genetically identical to what they could catch at the supermarket, because this applies to flu too, right? I give people flu viruses and they can go catch it, but I still worry about the patient that I gave the H1N1 going out and giving that H1N1 to someone else. And the reason I worry about that is because I actually gave that particular H1N1 to that person. I don't want them to go and infect someone else with the H1N1 that I gave, even though that other person could go out to the supermarket and catch it, or go get in a cab and catch it, or go on the bus, whatever. So I know it's a little ticky-tack, but there is kind of a difference between the virus that we as a researcher introduce to a person compared to something they caught in nature. So we're a little bit careful about that distinction. I can understand how that can be frustrating, but I think we do have to be a little careful because we are intentionally doing something to someone. So we do have to consider that and be very careful and make sure people are fully consented and that we do protect the community so that they don't catch what we introduced, even though it's the same thing that they could get on their own.
For me, Nadine, to come back to Matt about this. So you're totally right, Matt, that there is this difference that in many contexts matters a lot philosophically and in medicine between I introduce the risk versus it's a natural risk or somebody else introduced that to you. I feel that in this context, it shouldn't matter. So an example from clinical care, if I developed a stroke now, and I would be rushed to the hospital I'm looking at through my window, and they would verify that I'm developing an ischemic stroke. They would be tempted to give me tissue plasminogen activator to dissipate the blood clots that are causing the ischemic stroke. But that one comes with the risk of causing me hemorrhage, another risk. The guidance that we have since 1995 says, therefore, you should give it only so long within a window of 2 to 5 hours from the onset of stroke that the risks that you're introducing are smaller than the risks that you're preventing. If it were the case that we should put extra emphasis on not having done the risk, not having done the harm, the guidance should have been different. It should have been even earlier than 2 to 5 hours, because we put some weight on not having done that to you. We don't want our hands soiled. We should set aside what is best for you. I mean, from your point of view, it doesn't matter whether the disease, the stroke did it, the natural disease did it, or the doctors did it. We should not be the best advocates of your interests. We should rather stop short of doing what is maximizing your chances. We should instead put some weight on not having done any harm to you. And clearly that would have been a less good recommendation than the one we have now. We want to stick to the recommendation that says doctors should just try to be the best advocates for their patients. So I think in these contexts, we actually don't mind whether the risk comes from the doctor's actions or from the natural disease.
Yeah, I would just say about that you're still looking at two different scenarios. And this is why I don't remember. I was in graduate school when I heard this lecture, but it actually might have been the former Surgeon General Koop, who came and gave a lecture when I was in graduate school. I heard this idea that, a regular physician wears a white coat and that a researcher should wear a green coat, because a white coat doctor does exactly what you said. Their goal is the best interest of the patient, at least it should be. But me as a researcher, when I do research, although I'm cognizant of the safety and the issues regarding the individual, the goal of what I'm doing is not always in the absolute best interest of the individual, right? It's in the best interest of the goals of the research, the public at large, and so on and so forth. So when I'm taking a completely healthy person and I'm administering a virus to them, that's very different than a person who has a problem that needs treatment. And now I'm maybe taking a slightly risky treatment and giving it to them to deal with that problem, because what I'm doing is more like giving TPA to someone who's completely healthy and doesn't need it. Right?
Absolutely, Matt.
So,
I didn't mean to suggest
a little bit different there. So I want to be careful about that.
I totally agree on the green coat element. I only meant by the example to say, even in clinical care, where they're best advocates for the interest, and you're also totally right that, of course, we couldn't justify challenge trials or basically toxicity trials on healthy volunteers. Right. If the point were to serve the best interests of the individual medically.
Yeah.
Thank you all. This was a great discussion.
Point of view, as a person who might volunteer for a challenge trial, it doesn't matter to me. I figure I'm going to get exposed to Omicron regardless, and I would rather get exposed to it in the lab. So I make that choice.
Thank you for the input, and thanks. This was a great, lively discussion. I know we're over time, so I want to thank everybody who attended the call today. Many thanks to our panelists. Happy holidays, everybody. Stay safe.
Thank you very much, Nadine.
Thanks. Bye-bye.